It's so great to be back on Chestnut Chat. My name is Jared Westbrook. If you don't know me, I'm the Director of Science with the American Chestnut Foundation. And a few exciting things today. First off, we have a new CEO. His name is Michael. We're really glad to have him on board now. He's been working for us now for 3 weeks, and it's been great to have him. And with this Chestnut Chat, I just wanted to say that we're going to be planning to do these at least every other month. These first couple, we're going to have actually one next month as well. And these first two chats, we wanted to focus on, examples of successes with breeding. And the first one we're having is with Trevor Walker from North Carolina State University, who's going to be talking about breeding in loblolly pine, some things that are very similar to our breeding program, breeding for growth and disease resistance. And then the next chat in November is going to be Jessica Rukowski from the University of Illinois, who's going to be talking about basic concepts of recurrent selection and how that's worked in crop breeding for a long time. So these two kind of will give us some foundations about where we're going with our breeding program. But for now, I just wanted to introduce Michael. He'll introduce himself to you all, and then we'll introduce Trevor. So Michael, take it away.
Thank you, Jared. Thanks, everybody. Thanks for being here with us on Chestnut Chat today. It's exciting to see the numbers and the excitement over this conversation that we're about to have, which personally I am thrilled about because if you have had a chance to look at my background, I doubt you have. I'm a forester by training. So I grew up in Western New York State and grew up on the shores of Lake Erie and learned about the importance of forests and water quality and pursued a career in forestry and policy and trying to make the world a better place for forests and people. And that's really been what I've been about for the last 30 years or so. Coming to the Chestnut Foundation from a different foundation, the U.S. Endowment for Forestry and Communities was the last position I held before I did some work on a documentary film. But that position was really about trying to create new markets for forest products throughout the country, working on things like cellulose and nanomaterials and other innovations in forest products, really getting into the science of how we get there and how we make those next innovations, which is why I'm so excited about what we're going to hear about today. Because when I decided that the Chestnut Foundation was someplace that I wanted to come to and try to get this tree back in the ground, the reason that I'm so excited about it is because of our work of this new breeding program that we're going to talk about today. The work that has been done at North Carolina State University, the plan that Jared's put together with our team is exactly what we need to be doing to get this tree reestablished throughout the United States. If I look at NC State and what they accomplished with loblolly pine, it is really one of the most remarkable stories of breeding that no one knows about. The fact of the matter is we have 50% volume gains in pine trees in areas of the Southeast because of the work that's been done at NC State. That means a tree that, a few years ago was relatively small at year 7 is now available for pulp timber harvesting. That is the work that we're capable of doing. That's the work that we're going to do now with Chestnut. And I'm really excited to hear from Trevor to tell us about how we do this, how it works, why it works, the science behind it, and how we're going to be able to take your work, your volunteer work, and actually apply it to this model so that we can get this tree back in the ground. And I'm just excited to hear about everything we're doing. I'm sure we'll get a chance to talk more in the future. Please reach out to me. My email is michael.gergen@tacf.org. Look forward to chatting with you, but I really want to hear from Trevor. So thanks, everybody, and can't wait to hear this presentation.
Okay, cool. Thank you, Michael. And so today, pleased to introduce Trevor Walker. Trevor has been with the North Carolina State Tree Improvement Co-op as co-director since 2023. He's been working with Dr. Fikret Isik, and we joined the North Carolina State Tree Improvement Co-op this year, and Trevor and Fikret have already come out and visited the sites, seen some of our progeny tests both at Meadowview and in the forest, have been up to Lassane State Forest in Virginia. So Trevor has started to see like some of the stuff that we've been doing with TACF and has been very supportive of the recurrent genomic selection program. They're doing a lot of the similar stuff with loblolly pine at a larger scale. So it's just really good to have like peers and friends that are, making that have a lot of experience with this and have gone through various cycles of learning themselves, and things that have worked well and haven't worked as well. So we are so grateful for their support, and we are pursuing a lot of the similar things that they're doing at NC State, across the East Coast with working with forestry companies. So we invited Trevor because this kind of provides an example of a success with breeding, and something that we're looking to incorporate the techniques, much more rigorous in our program, at what they're doing now. So please take it away, Trevor.
Well, thanks a lot, Jared and Michael, for the introduction. I'm going to go ahead and share my PowerPoint. So a little bit about me. I also have a forestry background. I did my undergraduate in forest management in Texas, and I also studied for my master's there at Stephen F. Austin State University. I studied forest accounting, you might call it, but it's really biometrics. It's quantifying, taking measurements of trees and then using those measurements to make decisions. And then I got introduced to genetics after I'd already established a career in forestry and been working for industry. I wanted to go back to school, get my PhD, and I was introduced to genetics, quantitative genetics. And it was just a whole new dimension of forestry that I really it was a challenge for me to learn, and I'm still learning more about it. But it's such an awesome field of study because it's really the frontier of where we are in forestry in my mind. I've really enjoyed learning about the American Chestnut Foundation over this last year and a half, interacting with Jared and Vasily and others with their team. And I'm glad that we have this partnership now. Let me first take a step back and talk about loblolly pine. So loblolly pine is an incredibly important timber species for the southern United States. The economic impact of this species is really huge. About every year, about 1 million acres are planted in the southern United States with this species. These are typically reforestation sites. So you have a tract of land that a landowner has sold his timber and he wants to plant back some trees so that forest land stays productive. And every year it's about 1 million acres. It's about 800 million seedlings. Sometimes it's as many as 1 billion loblolly pine seedlings getting planted every single year. And what is really impressive about those is 95+% of those seedlings have some level of genetic improvement. I'm talking about artificial selection for improved growth, straightness, disease resistance. And I'm really proud that probably 75% of that material comes from the program at NC State University. This program I'm immensely proud to be a part of because it's been around so long. It was founded in 1956 by a visionary tree breeder named Bruce Sobel. Bruce went on to found other tree improvement programs. He also founded an international program that's housed at NC State University. He was a professor at NC State University for a very long tenure. And he had contemporaries like Norman Borlaug, folks that used the concepts of genetics to really save nations from starvation. So trees are very important. You can't eat a tree. It's not going to save you from starving. But whenever Bruce was starting this program, the forestry outlook was very different than it was today. This was post-World War II. The United States economy was booming. Most of the timber, a lot of timber was being cut for newsprint, paper, for lumber, a lot of new housing starts, suburbs being built across the nation, especially in the South. And most of the timber that was being harvested was coming from fallow ag fields after the Great Depression. During the Great Depression, we had a lot of ag fields seed in, go fallow, and that's where most of the timber was coming. And it was not hard to see that it was not a sustainable source of wood. for the booming economy. If the consumption grew at the rate it did, foresters are going to have to do something different to make sure we don't run out of wood. And so Bruce, he immediately recognized the importance of genetics in helping to solve that issue. Lots of other research co-ops at universities across the United States looked at other things like silviculture, how to prepare the land using herbicides or fertilizers where needed, and how to thin stands to improve forest productivity. This is really the forestry, especially the South where I'm familiar, the research and the university cooperatives played a big part with their industry partners to making forestry sustainable in the region. Our particular program, it includes seedling companies and state agencies. This is who funds us. This is who keeps us going day to day. Also timberland managers that are planting. And we have a class research associate members. These folks may not be very interested in loblolly pine, but they're interested in the methods that we're using for genetic improvement. So we have international organizations in South America and Canada. And the Chestnut Foundation is a research associate member. So our present focus is really on loblolly pine. We've got some work with longleaf pine, but most of our resources are with loblolly pine. So this is just a big list of all of our members, our present members. What I want to draw your attention to are these folks that are full members. If you're in the South, if you're planting seedlings, you may recognize some of these folks. Some of these folks are all over the country and even international. These folks don't just fund our program. They help us by planting test trees, by breeding trees. They coordinate with us on a day-to-day basis to keep the genetic improvement going. Our staff in Raleigh, we have 4 research associate staff members. We have 2 full-time professors and then 2 professor emeriti, and breeding specialists, administrative help, and then what's really important is our graduate students. So our graduate students help us. They motivate us to keep a very strong research culture so that we're researching new technologies, because these students, they have to have exciting research projects to publish on to get their degrees. So what exactly do we do? I've said a little bit about who we are, but what are we doing? First and foremost, our bread and butter is managing the breeding program for loblolly pine in the southeastern United States. And this is all about developing new genotypes that are going to be used for reforestation. I'll talk a lot about that. Another mission that we have is research, right? We're at a university. If I don't do good research, I'll get cut from the faculty. And my graduate students won't have awesome things to publish on. So we have a very strong research culture so that we develop new technology, new methods. We also have a very strong education mission. So I teach courses. My colleague, Dr. Fikra Issac, teaches courses. We do a lot of workshops and extension events with our members. And we also have this last category here. It's called performance rating system. This is a little bit newer. This is a system that we've put forth to publish these genetic scores so that the common landowner can understand them. We take our big data analysis and we summarize it to help folks that are planting seedlings, buying seedlings, growing seedlings, help tree farmers make good decisions. And I'm not going to talk about that today, but if you have any questions about it, please feel free to send me an email. So this diagram really shows what it's all about for making genetic gain in a tree improvement program. So the basic concepts of recurrent selection, that's what this process shows, is taking individuals that look good. So this is an individual tree that has all the traits we want. It's tall, it's got nice girth, it has no diseases, and it's straight as an arrow. We take individuals like this, what we actually do is we take clippings from the top of them and we graft them into a breeding facility so that we can make crosses with them. We can't really make crosses with this tree in the woods because you can't get a bucket lift out there. We need to clone it and take it somewhere else. And lucky for us, Loblolly pine clones through grafting very easily. So once we get it into a breeding facility, now we make controlled crosses. What we do is we make new genotypes that have never been made before by crossing, using controlled pollination to cross trees that otherwise would have been very far apart on the landscape, would not have had an opportunity to mate. We'll make a controlled pollination to make them. Once we get the seed from those, we plant them in tests. And these progeny tests are crucial. They've got statistical designs to help us determine which trees have good genes, which trees don't have good genes. And testing is crucial, right? You choose it, you see a tree like this in the woods, and it may look great, but a tree is a product of its environment. About 80% of what you see, 80% of the differences tree to tree is because of differences in their growing environment. Subtle differences in the depth, the soil depth, right? Depth to some hard layer, subtle differences in the amount of sunlight they receive. Only about 20% of those differences are due to genetic factors. And so when we pick a tree that looks good, we might have reason to believe it's because of its genes, but we have to test it to validate it. So this is not a new technology. This is basically caveman technology, this recurrent selection. Native Americans fundamentally changed this dumpy teosinte plant into modern corn using these methods, right? These methods, dogs were domesticated from wolves with this approach. And it's been used in pretty much every aspect of food production, livestock, poultry shown here, in plant breeding. And it is a commonplace. Pretty much every advanced forestry region in the world has an active tree improvement program to make sure that their forestry is sustainable. Here's what our program has done. So on the left is a row of what we call wild, non-improved, no artificial selection, just the way pine trees were growing whenever forestry was really getting started in the South. And then on the right is an improved family. The only difference between these two is we know the parents of this tree right here. We actually just know the female parent. It's an open pollinated family. We know the female parent, and we tested it, so we knew it would be a good parent. Here's a similar photo. This is at 5 years old. These trees are 5 years old in Florida. On the right is probably what is representative of what folks were planting in the 80s and early 90s. And on the left is representative of what folks were planting in the mid 2000s, 2005 and 2010. So if we look at our data, take a retrospective look, how much gain have we made? How much genetic gain have we made by spinning that cycle around? This graph, it shows a box plot. This is the distribution of a handful of wild, non-improved, we call them check plots, but there's about 5 of them, and then this is their distribution for productivity. The y-axis is the volume of the tree at about age 4 or 5. We measure them as young as we can. I'll explain that soon. When they're about 4 or 5 years old, the average wild non-improved is going to have about 0.4, 0.5 cubic feet of volume. So not a lot of volume, but with recurrent selection, we've been able to improve it cycle over cycle. And so this is a distribution of trees, performance of trees in our program, because we have some that are excellent growers, some that are not as good, but much better than where we started. And a recurrent selection program really depends on maintaining a diverse population so that you can continue getting those gains. We could maximize gain from one cycle to the next if we just pick the best 2 trees, but then we'd run into a wall in the next cycle. And that's one of the challenges of that's probably the primary challenge of tree breeding, is that balance on gain and diversity so that you can make long-term gain. So really great gains for productivity. Our species, Loblolly pine, also is affected by a disease. It's called fusiform rust disease. So this box plot shows the rust disease over the cycles. And this is the distribution of percent of trees that are infected on average. So if you go look at wild non-improved, on average, they get between 30 to 40%, 45% infection. Through our breeding, we've been able to bring down that average considerably. Now you may be looking at cycle 4 and cycle 5 and some of these and saying, well, hold on a second. These you didn't make as much gain from that cycle. And it's a nuance of breeding that we want to make gain for multiple traits, productivity, and rust. And it turns out the ones that are best for productivity are not necessarily the best for disease resistance. That's why we have a breeding program, so we can shuffle those genes into new combinations and try to select trees that are great for all those traits. Finally, stem straightness is another trait that we're working on. In this metric, lower values indicate straighter trees. We made a huge jump with our very first round of selections. We fundamentally changed the species. Loblolly pine is not naturally a straight species, but in that first round of selection, an incredible amount of emphasis was placed on straightness, and it turned out those straight trees that were chosen were straight because of their genetics, not just their environment. So we made a lot of gain in the very first round of selection. We haven't made as much gain recently because we've been putting more emphasis on productivity and disease resistance. In the last cycle, our emphasis was 60% growth, 20% rust, and 20% straightness. We're rethinking that for our next cycle, and the cooperative members are requesting, hey, maybe we should put some more emphasis on disease resistance and straightness, back off the growth. Now, I'd love to do 100% growth, 100% rust, 100% straightness, but it just doesn't work that way. You have to pick and choose. So we're spinning this cycle to make gain. I want to go back in time to what the very first selections looked like. So when this program started in the 50s and the 60s, the genetic base were wild natural trees across the landscape. And at that time, each company that took a part in founding this program, not just companies, also state agencies, had foresters scour the woods for perfect trees, ideal looking trees. We call them plus trees. They're naturally occurring, but they look phenomenal. They've got all the traits that you would want for whatever your objectives are. So to get the program going, they got these plus trees and they grafted them into the breeding facilities so that they could mate them. This map shows where the plus trees first originated from, and they did a really good job of covering the South. You'll notice this area right here skipped over. That's slash pine country right there. Slash pine had its own improvement program, but for loblolly pine, they did a good job of covering the natural range. Each dot represents where a plus tree was selected from. And then this graph right here, it shows our pedigree. So the red dots in this map, those are still contributing to our breeding program. We've since culled the white dots. We decided that the tree from that county was not of genetic merit to contribute to our breeding program. So that's what this pedigree shows. So the top represents the trees that founded our program. It was about 4,000 trees were used to start the program. But if you look down here, this is our 5th cycle population that we're mating right now. Only about 500 of the original 5,000 trees still have genes contributing to our breeding population. So nowadays, we don't go to the woods to select natural trees for our breeding program. We use our progeny tests. Our progeny tests are so important because they've got a statistical design that helps us determine if a tree looks good because of its environment or if the tree looks good because of its genes. We know the mother and father of all these, and we can get an estimate of that tree's, we call it the breeding value. It's the genetic merit of that tree. Now it has a standard error. We still have to test the tree. And how do we test it? So the way that you test a tree to see if it has good genes is you collect cones from it and you plant those seed. Because whenever you plant a lot of seed from a tree next to seed from other trees, you build statistical replication so that you can say with confidence, hey, all the seed from this tree they make great seedlings, they perform really well. I've got confidence that it has good genes and it needs to continue in our breeding program. So select the trees, graft it to our breeding facility so that we can make crosses with those. This picture shows our breeding facility. It's located in central Georgia. We're very fortunate that the Georgia Forestry Commission hosts us. It's their facility, but they've given us access to do the breeding there. And what you see here are rows and columns of trees that are basically acting as rootstocks. They are sitting there waiting for us to graft into them so that we can do the mating. Whenever we graft the trees, an important technology that we have is called top grafting. We like to graft them about 30 feet in the air. And so that's what this lady is doing. She is grafting a new selection, a tree that's been selected to participate in the breeding program for the first time, and she's grafting it way up high. She's in a bucket lift. The reason why we like to graft them way up high, you see this tree on the right, it was grafted right here where it's been pruned back. We like to graft them way up high because it stimulates flowering. It makes them flower sooner. If you were to graft them on a tree that was short down here, it might take 4 or 5 years to make flowers. And that's a problem. That's a big problem because the sooner we can spin that cycle that I've been showing, the faster that we can turn a cycle over, the more gain we can accumulate per unit time. So turning that cycle as fast as we can, being organized, doing things like grafting in the tops of trees to induce flowering. Those are technologies that we use to accumulate genetic gain as effectively as we can. So once we've made crosses, we do testing. We got to test those seeds to make sure that they really do have good genes. And this is really where our cooperative network is so important. If you remember the second slide I showed, it showed our full members. There's about 10 of them. These are forestry companies and state agencies. And the program could not be successful without them because they plant so many progeny tests. Every year they plant about, as a group, they plant 10 tests. So this figure just shows the tests that were planted during our fourth cycle between 2014 and 2022. This gives us the data we need to determine which trees have good genes for which traits and help us decide which trees we're going to mate and ultimately which trees get planted on the landscape for reforestation. So this is what a typical test looks like when it's being measured. The trees are only 4 to 6 years old. We want to measure them as soon as we can. Now, we don't measure them when they're 2 years old because if you measure them when they're 2 years old, right, you might have a tree that's taller than its neighbors, probably because maybe it just got planted a little bit nicer. Maybe one of the neighbor trees got bit by a deer. There's a lot of environmental differences. With repeated measurements, we found out that about between ages 4 and 6, that's whenever we can get a good idea of if a tree looks good or if a tree looks bad, it's because of their genetics. So what do we measure? We measure how tall they are, how much girth they have. We measure if they have a fork, yes or no. We measure how straight they are, and we measure their disease if they're infected with the disease. So this technology is not, we've made incredible gains with this technology, and it's really pretty basic. We've done a good job at it because we've been organized, we've been persistent, but at its basic, it's pretty simple, nothing earth-shattering here when it comes to the technology being used. Our group, as the research mission of our group, we've looked at multiple advances in DNA, genotyping, sequencing. We've seen what folks have done with biotechnology and agricultural crops. in livestock. And we followed those things closely. We've done many research projects to implement their feasibility. So trees are difficult. Trees, they have to be on the landscape for a long time. For a lot of conifers, they have big genomes that are repetitive, and it's hard to find your way around. So, and whenever we try to associate DNA markers, individual DNA markers with traits, a lot of times we don't find anything because, right, a tree can be tall for due to many genes that are affecting it. It's not typically like, this tree got the grow fast gene and this tree got the grow slow gene, right? It's a many genes that influence why a tree grows fast or grows straight. But there is one technology that we started researching about 10 years ago. We started studying it with intensively, and I'm really excited to say that we've prepared to implement it in our next year of selection. So I mentioned earlier that spinning the cycle as fast as we can is crucial to getting our genetic gains as quickly as possible so that we can compound those gains. If you look at the bottlenecks here, there's a big bottleneck in the time it takes between you plant these trees and you measure those trees. If we could bypass that, we could very much speed up the cycle. And so instead of selecting the trees when they're big, there's a new technology that came out in the last 10 years, 15 years, pioneered by livestock breeders, that allows you to select the trees when they're seedlings, when they're this big, versus waiting for them to grow up to measure them. It's called genomic selection. And the way that it works is you have a population of trees, progeny tests, that you've measured. You've measured all the traits you want, how tall they are, if they have a disease or not. And then you've also genotyped them using a panel of thousands of markers. Now, these markers themselves don't tell you anything individually. They're just measurements of what genetic variants you have, and they're really good at telling how related two trees are. It's the same technology that Ancestry.com uses, 23andMe uses. They're really good at saying, hey, these two trees are really related, or these two trees, they're not so related. Well, with that technology and a rigorous training population, you can build a model that predicts the breeding values as a function of these markers, thousands of markers. You can apply that model to very young seedlings and make your selections whenever they're too young to measure. So in this manner, we can, after we do the breeding, when the trees are small like this, we can genotype them and then make the selections without waiting for them to grow up to size to actually see if they're a good looking tree. So we initiated a project in 2017. We started the breeding and the trees were planted in 2021 to see, okay, does it work? And I know this graph is a little bit busy, but if we look at the left two histograms up here, this represents what we had been doing, waiting for the trees to get 4 years old. You've got this generation 1 distribution in red, and then generation 2 distribution in blue. And if you just wait for the trees to be 4 years old, 5 years old to measure them, we see, hey, yeah, we made gain whenever we bred that population. Well, we can repeat the experiment, but this time we can pretend, hey, let's pretend like we selected the trees when they were this old, when they were only 1 year old in the greenhouse. Do we still get the same outcome? And what we find is those markers do a great job of predicting how the trees are going to perform. Caveat, as long as you have a good training population. So going back to this slide, as long as this population is a rigorous population, this model can work really well. Now, what happens if it's not a good training population? That's what this graph shows. So what makes a good training population is that it's related to the population that you're predicting. They have strong pedigree relationships, genetic relationships. If you build a model and then you take it and you try to predict it on a new population, maybe built your model using pine trees in North Carolina, and then you applied it to pine trees in Texas, it's not going to work at all because there's no relatedness between those. So we measure the predictive ability using a value. It goes from -1 to 1. And we see really good results. And it's not perfect, but we see results that say, hey, we can skip that testing phase as long as we have a training population that's very strongly related to our validation population. So we're spinning the cycle around. We're in the next year or so, we're going to try to skip this part so that we can make the selections earlier. Now, we're still going to test. We have to test, right? We have to rebuild this model every generation to make sure that it's up to date. But what we're going to be doing is we're going to skip this testing phase when we make our selections so that we can spin the cycle faster. So we're doing this work. How does this actually get to the landowner? And the way that we do that is through what are called seed orchards. Now, seed orchards look different than breeding orchards. They've got multiple copies of the same genotypes, and their goal is to produce lots of cones for deployment across the landscape. So I'm really proud of this graph. The cooperative members started submitting survey data on how much seed they were producing from their seed orchards. And they've done it every year since the program's inception. And you can see how the gain has gotten out to the landscape through these seed orchards. So first generation orchards peaked in the mid 80s, and second generations really peaked in the early 2000s. Now fourth cycle orchards are starting to come online. Even though we're in our fifth cycle breeding, there's a time lag before it gets deployed onto the landscape. And anything we can do to reduce that timeline is gonna improve forest productivity outcomes. Okay, so now I'm going to talk a little bit about the disease that we work with, right? Chestnut folks, I know y'all are very interested in what we can do to improve disease resistance. Now, the disease that affects loblolly pine is a little different than chestnut blight. So both the host, the loblolly pine, and the disease, fusiform rust, are native. And they've co-evolved, this pathosystem has co-evolved over hundreds of thousands of years in this region. Now they've migrated with glacial incursion and retreat, but they've co-evolved with each other, and the disease needs the pine tree to be successful. So the life cycle is pretty interesting in that there's a phase where for the rust to complete its life cycle, it has to germinate on oak leaves. And so if a pine tree is infected, it can't infect another pine tree. The disease has to travel to oak leaves to complete part of its life cycle before it can travel to the pine tree. So here is an example of the disease affecting some trees in a genetic test. Unless this picture's pretty old, early on it was discovered that there was genetic variation in resistance. So here's another family of pine trees where none of them have been affected. So we can do something about it with selection. If you look at the hazard of the disease, it's very prevalent across the landscape. As you go north, it's restricted by cold weather, but the probability of infection tends to have hot spots. But for the most part, there's hazard throughout the region. And it is pretty devastating. This is some work one of my students did looking at the effect of survival as trees grow based on the hazard of the disease. So if the hazard is at 50%, you can expect almost 30% of your trees to die by the time the stand is 10 years old. So it's very crucial for improving forest productivity. Now, this co-evolved system, the disease and the host, they've sort of been fighting this battle with each other for millennia. And whenever you look at resistance, we know there's genetic resistance out there. When you look at it on the landscape, this is how it tends to play out. So you might have a lineage of pine trees that are particularly resistant, maybe like that photo I showed earlier. a lot of times you can identify 1, 2, maybe 3 genes within that family, within that lineage, that are very much responsible for the resistance. Using genetic mapping techniques, you can find a few genes that are deliver major gene resistance. But if you take a step back and look at the whole population, maybe there's another family that's also resistant in a different state. It might have major gene resistance, a few genes contributing, but it's probably different genes, because those two lineages, they evolved in different environments. They've separated geographically. Further, the virulence of the disease has also been evolving, right? As pine trees evolve resistance, the disease is an arms race for it to survive, to develop virulence. And so we find that different races of the pathogen across the region have different virulence genes. So the way that this works, the way that this manifests, is that within a particular pine tree, you can find major genes for resistance, which breeders love that, right? That means that we can make lots of gain. But it's only, those genes are only for a particular lineage. When you take a step back and look at the whole population, it's many genes. Many genes makes it more complicated. Many genes means you need to focus on using recurrent selection to improve the population. So this work, a lot of this work was discovered through lots of progeny testing, with artificial screening at the U.S. Forest Service Resistance Screening Center in Asheville, North Carolina. That's how we learned a lot of these hypotheses. I'm going to briefly share some work that a graduate student in the program did, Eddie Lauer, because I think it demonstrates this phenomena of major genes versus population level resistance. So what he did is he took one family. He knew that a parent tree tended to have resistance based on progeny test data, based on those field sites that our members are planting. He looked at the data analysis. He said, wow, this family is pretty resistant. Not every tree is immune, but some trees get infected, but overall the percent infection is very low. So he did is he crossed it with a tree that was very susceptible. Now this experiment's been done with chestnut, American by Chinese, almost an identical experiment. But for this particular family, what he found was there were 2 genes on the same chromosome that were very much conferring resistance. They were highly associated with the disease presence. And so what we did was we initiated a project to stack these genes into a using breeding to make new combinations to create individuals that are carriers for both of these big R resistance genes. This is what we're looking for. This is a pretty cool research experiment. This is awesome. We're going, with this work, we plan to develop a family of pine trees. It looks like they may be immune to the disease. However, that's not the end game, because we can't reforest the entire South with one family of pine trees. Further, anything that we cross with this to get the resistance genes, with breeding, you get half the genome. You get half the genes from your mom, half the genes from your dad. We can get these into new families, but they're going to be, our pedigree is going to be dominated by this particular parent, by this particular lineage. That has consequences for our long-term gain, right? We can run into a wall with our long-term gain. It has consequences for potential for inbreeding. And so while we can implement this major gene resistance for a particular family, for managing a breeding population, it's not going to give us sustainable gain. And that's why at the population level, we just manage it using recurrent selection, which treats it as if many genes are contributing to the disease resistance. So I want to, the reason why I tell that story is because it comes up a lot on the idea of using newer technology to improve the way that we breed trees. And this book that I'm showing here, it's sort of a memoir by Dr. Bruce Sobel, the founder of our program. It really details how the tree improvement program and other companies came together, other state agencies came together to accomplish the work that I presented on today. And the memoir, in the last chapter, right as Dr. Zobel was retiring, biotechnology was, we were making, the genetics and genomics community was making great advances in biotechnology. And, you know, Dr. Zobel was late in his career at this time. He met it with a bit of skepticism, but if you read the last chapter in this book, you'll see that his approach was very wise. He states he has no doubt that in the future, we're going to be using these new technologies, either gene editing technologies or marker-assisted selection technologies. We're going to be using them regularly. But breeding is still important. And he lays out some concerns that as he saw some companies moving their investments away from the breeding program into the biotechnology program, that probably will have undermine, that would likely undermine their investment. And so to this day, we've made great gains in loblolly pine, and 0% of the loblolly pines planted across the landscape have been the result of breeding using genomic technologies, biotechnologies. It's all been traditional selection. Now, we are working hard to incorporate new technology. I think in 10 years, that won't be the case, but let's not, forget about the gains that we've made so far. And so that's Bruce Zobel's message was continue the breeding and, invest in the research. Research is critical, but don't forget about the breeding program because it's tried and true. So I'm going to take a retrospective look. I've really trumped up our program and shown you a lot of good things that we've done. But we've also learned some hard lessons. So thinking about what could we have done better? If you look at the first and second cycles, a period of almost 50 years, we could have made a lot more gain. These two cycles were characterized by incredible investment, but not very efficient use of resources. And ultimately, they were too diverse. They had too many trees in their program. Remember, I said we started with 5,000. Now our program only has lineages from about 500 of those, and we'll likely further cull it because there was just too much diversity, and it was holding us back. The third cycle, it was a good plan put forth using the technology that was available, but it ignored some very important statistical advances that were developed by livestock breeders, particularly the BLUP and what's called optimal contribution selection for smarter mating designs, more efficient mating designs. The third cycle strategy was using, it wasn't much different from the first cycle, and it was really just dragging on. So some good choices were the third cycle strategy. It was never completed. The program did not complete their third cycle. They abandoned it and switched over to the fourth cycle. And I will say a good choice, I've said it before, but while we've been researching biotechnology with research projects like I presented, we haven't ignored our breeding program because it takes many resources with lots of partners planting tests, but it's effective. So I'm really excited about this collaboration that we have with the American Chestnut Foundation. Dr. Isick is on the science advisory board, and he's the one that really started, talking about, hey, we should concrete this collaboration. And so I've really enjoyed learning about chestnut over the last year and a half. What I've presented on fusiform rust disease and loblolly pine, right? It's not the same thing as American chestnut and the chestnut blight. Chestnut blight is an exotic species that did not encounter major gene resistance in the natural population. So there's differences in the biology of the pathosystems for sure, but there's also some things that I think are important overlap. And one is the need for progeny testing. Another is the need for continued breeding. and treating the trait as a at the population level as being controlled by many genes. Just using recurrent selection to test families and to cross individuals that they may not be perfect, they may not be immune, but they show more resistance compared to the population mean. That's what breeding is all about, right? There wasn't just one day, when the cavemen didn't just one day find a wolf that looked like a dog and said, all right, let's keep this one, right? It was a slow process of wolves that started hanging around the campfire, were more receptive to human interaction, that eventually created man's best friend. So that's the way that we've treated breeding in our program is slow and steady wins the race. But also really thinking about the things we can do for our particular species, like top grafting, and then in implementing new technology where it has a high probability of success, like genomic selection. So that's all the material I prepared. I would be excited to take any questions.
Thank you, Trevor. Really good to hear about the history of NC State Co-op. So I see there's a couple questions. If you guys have a few more, I'd love to hear them. I have some questions myself. So one of the things that we often we get a question about, we are going to be breeding for restoration. So we're trying to improve the population, and but the tree, like with the Loblolly pine, you have lots of variation within families. You're going to get, like you're doing this very controlled breeding. We are going to be improving the population through this breeding cycle, but then what happens with restoration, where we will be planting open pollinated material from our selected parents? Like there's going to be a lot of variation in that population. And so how do you think that, like, will the population, with enough improvement, will there be enough, I guess, natural selection to maintain those gains in the population over time? Because ultimately we want to get a self-sustaining population of trees. And what you brought up, the example of um, dogs and wolves, you don't see dogs going back to wolves if you just let them breed on their own. So I guess, what do you think about, like, how this gets into the wild, and what happens if you were to, let's say, just hypothetical, what happens if you were to just allow, you allow these pine trees to improved stock to breed with each other on the landscape? Are their progeny just naturally much improved in their growth? Have you seen any of that? What happens in the wild, I guess, is the question.
Yeah, what you're describing to me sounds a lot like what breeders call a land race, where the concept of a land race is you take seed. It's typically whenever a new species is introduced to a new country for agriculture production, and maybe maize from Mexico. A great example is maize originating in Mexico was being brought to the Midwest to get planted, and a big constraint was flowering time. The maize would want to wait too late into the season to flower because it's used to living in Mexico. But after a few generations of selecting those maize plants that tended to flower a little bit earlier in the Midwest, they're able to develop a land race. So that's the first thing that comes to mind. The second thing is just really underscoring the need for progeny testing, for field testing. That is where the gains, that's where you test the material. Now, this resistance, it has to be durable. These trees have to survive on their own in harsh weather conditions. It's not just being disease resistant. You also have to be adapted to your environment. And chestnut from northern Alabama versus chestnut in West Virginia, those are going to have different adaptive requirements. And so having a diverse population coupled with progeny testing to understand that adaptive variation that's needed, that's the kind of data you really need to answer the question.
Thank you. I have a question from Greg Ross. and it's related to, I mean, we have a arm pursuing research in biotechnology, understanding the mechanisms of blight resistance, doing the genome sequencing, comparing the Chinese and the American, seeing what's different and why one species has evolved that resistance and American chestnut susceptible. Um, do you see, I mean, what have you seen over time with the kind of work and research into biotechnology in Loblolly pine, where you've had, like, for example, ArborGen, that company in South Carolina, based in South Carolina, has done a lot of research into biotechnology and transgenics and things like that. And like, what was the outcome of that? And is there still a desire for doing genetic engineering in loblolly pine? Does it have a place in the future, do you think, for improving
Absolutely.
these traits that you're interested in?
I, absolutely right. Every technology should be on the table. The path for genetic engineering, though, is it's a long path. And it requires time, investment, and resources. And it's going to start with a reference genome assembly. Right now, we don't have a reference genome for loblolly pine. What that means is that if you were to get sequence data from a pine tree, you don't know what chromosome that sequence goes on because there's no map, there's no road map. So first we have to get the reference assembly down. There's some really exciting work done at University of Florida to get us closer there. But right now, instead of having 23 chromosomes like we do for humans, the reference assembly for loblolly pine is 14 million contigs. And we don't know how they go together. So we need that first. Next thing we need is tissue culture. So once we have a reference genome and we find a gene that we want to edit, you got to propagate that, right? The way that it's typically a cellular level gene transformation, you're going to have to turn that cell into thousands of trees for reforestation. That's not an easy thing to do with conifers in particular. But even before you get there, you have to, once you have a reference genome assembly, you have to figure out what genes do what. And that's probably the biggest limitation, even for crops that are minimal to genetic engineering, maize, rice. The types of traits that you see improving aren't growth traits. Growth traits are controlled by so many genes. The type of traits that you see being improved are things like introducing herbicide resistance, something that wasn't already there. Introducing production of a vitamin that will make your population the population you're feeding more have more nutrition for the population that you're feeding. For loblolly pine, we have not annotated a single gene. I mean, we've looked at homologs with Arabidopsis and other species and say this is probably what this sequence does, but none of them have been validated because we don't have the reference genome. So getting the reference genome, annotating and validating the genes, getting our tissue culture together, those are the steps that's going to take. And if you stop breeding and put all your investment in those, you're going to miss out on a lot of forest productivity. So you have to do both together and meet in the middle. And, finally, the genetic engineering, it's just a tool. It can't be the save all answer because at the end of the day, a genetic transformation happens on a single plant. And we have a population of trees, right? For long-term gain, for maintaining adaptability and diversity on the landscape, we have a population of trees. So you're going to have to port that thing up to affect the whole population. And it's either done by many transformations, which will be expensive, or it's done by thousands of years of breeding and natural selection.
Yeah, the bottleneck definitely is that we still end up having to test it in the landscape, and that just takes time. And like single genes have limited efficacy. You're not going to just knock it. You're not going to, like you're saying, like find a super growth gene. Like that's why you're doing this recurrent selection, is you're combining a lot of alleles that you don't really know what they are and what they're doing, but they're making, you're making gains nevertheless. And single gene stuff with biotech, it's like still limited, and like we don't understand the networks that are involved in tree growth or cow making milk or chestnut blight. We have dozens of regions of the genome that are involved that we know, but like we don't know exactly in those regions what are the individual causal variants that are contributing to the trait. So we end up doing a lot of trial and error, and you still have to do testing. So it ends up being a major long term. Not to say we're not doing it, but it's just we're doing the breeding. Like I'm not going to like do the other. You're not going to sacrifice the breeding to do that. So yeah, I
The
totally
beauty
agree
of
on
breeding is
that.
that you don't need to know, what individual genes do. You just know that you work on the concept of resemblance among relatives, and that's how you make genetic gain. But don't get me wrong, understanding those will be crucial to making good improvements. But we can't get distracted.
Yeah. Okay. So curious. There a number of people are very interested in having the pure American chestnut. And so having blight resistant pure American chestnuts. And or we call them wild type. And there's been the history of another program. We've done this too. We've had these so-called large surviving American chestnuts. These are defined as trees that have had the blight probably for decades. They've lived through the original epidemic of the blight that came through on the East Coast, and they have surviving main stems, and they are some of them, the cankers, the whole tree is one giant canker basically, but it is alive. And so, what, like with that kind of resistance, it's possible to do a recurrent selection program, I imagine. So kind of what do you think about that? Like, how much gain do you think we can make with that? Like relative to hybrids, there's hybrids in pine, for example, hybrids in eucalyptus. Like those hybrids have been a part of forestry in production forestry as well, right?
Yeah.
So kind of talk a little bit about that. Like, would how do you feel about like the mixture of doing the breeding just within the American chestnut versus like doing hybrid breeding to improve the trait?
Yeah, absolutely. So hybrid breeding is done with most forestry species, typically do just like you're doing with chestnut to introduce a new trait. For other parts of the world, with pine hybrids, usually one species is more cold tolerant than the other, and these forestry companies operating just above tropical areas, they want to bring in that cold hardiness. It's just like you bring in disease resistant with the chestnut. Now, cold hardiness, right? That's not controlled by a single gene. So it's going to be controlled by lots of genes that, monitor turgor pressure and seasonality, when to break bud, when to set bud in response to photo period. So what those hybrid programs typically do is they don't continue recurrent selection with the hybrid population. I mean, some have, but a lot of them focus on the main species, improving the main species, and then just making the hybrid when they're ready to plant their trees. Our program, has never done that approach. We've always worked with the native species. Whenever we hybridize loblolly pine with another species like shortleaf pine, you basically get something in the middle, which has the drought tolerance, and shortleaf pine has rust disease resistance more so than loblolly, but it grows slower. So you end up with a rust resistant, slow growing tree. And that can be a non-starter for a lot of tree farmers. So that's why it hasn't taken off there. When it comes to breeding long-standing survivors, that's essentially what we did. When we started, we scoured the southeast looking for trees that were geographically apart. They look good. We suspected they had good genes, but certainly they didn't have the same good genes. And so we brought them together into a common environment, into our breeding facilities, and we made novel genotypes by crossing those. And I see that crossing those, the natural resistance in the program is going to be an important way if you're dead set on maintaining 100% American lineage. Um, if you're going to bring in some other uh, uh, Chinese lineage, that can also be a part of it too. You just want to make sure that the base that you're starting with is broad, and that the trait ultimately you're selecting towards what you want: tall, thriving chestnut trees um, over generations. So we use a tool to manage relatedness in our program. Jared, I know you're familiar with it. It's introduced by livestock breeders, and it helps us navigate that gain versus diversity trade-off. And it balances long-term gain with short-term gain. You know, tools like that, they they tell you what to cross. And so sometimes you look at the solution and you say, well, I don't want to do it like that. Why are you crossing that tree 30 times? Normally we only cross a tree 3 or 4 times. But you told it the balance of gain and diversity that you wanted, and it gave you the optimal solution for that gain and diversity. So I don't know the answer, but I know that using these tools and the data is how we're going to get closer to finding the right answer.
Yeah, just to follow up on that, we this year, this is a question from Paul. He asked about the large surviving American chestnuts. This year we have crossed we made maybe a dozen crosses with these trees. And next year we're going to be inoculating them with chestnut blight as seedlings, at least some of the progeny from those. And we're going to compare the resistance level that we see in those trees relative to the hybrids that we have. And basically what we're looking for is any families that have above the American chestnut, the baseline susceptible American chestnut mean. Those are going to advance in the breeding program, or we're going to plant out their progeny in progeny tests like what you guys have done at NC State. Probably will have those at sites. We'll have Chinese controls in there. We'll have susceptible American controls in the progeny test so we can show where these trees are on that spectrum of resistance. Eventually, we'll cut out those trees and we'll inoculate the trees with blight in the field when they're older and pick out the most resistant trees. So we'll have kind of a recurrent selection program going for the large surviving American chestnuts as a sort of a separate track. And we're going to continue with the hybrid breeding, especially for Phytophthora resistance, which is something that we have not seen in the South, in the populations in the American chestnuts. We still need that in the South. So for sure, we need baseline. We need that Phytophthora resistance trait that only we know comes from the Chinese so far. And we want to grow in the hybrids, tall growing, blight resistant trees.
Yeah.
So we're doing recurrent selection. So we're doing both, is the answer to that question. So.
But
Yeah.
the goal of the breeder is you have this population of trees and they're not perfect. But they all have, each one of them has some good genes in them. Now, if you got some trees in your population that don't have any good genes, you need to get rid of those. When I say good genes, we don't actually know what they are, but based on their phenotypes, based on progeny testing, they perform, say, better than average. And it needs to be a diverse population with lots of genes to choose from. The challenge is you got to shift those alleles, the copies of those genes, into a more uniform population that just has the good ones and leave behind the bad ones. And you can't do it in one cycle. It takes multiple combinations of breeding to keep shuffling that deck of cards to try to make good hands and then throwing away bad hands and reshuffling the deck of cards as an analogy. So I think that's an appropriate approach. The more trees you have in your program, that was our folly with the first and second cycles. Thousands of trees in our program took long time to spin the cycle, and they gave up a lot of gain based on that. So you want to have a diverse population, but if it's too diverse, then it's going to take more time to get the genes out. And that's the challenge of the breeder.
So one question from John Welker is, have there been any studies on the genetics of the pathogen, Crenartium? What was the other one? Pitch canker. What is that one called? Do you guys have pitch canker and loblolly? Is that a problem?
Yeah, we see it occasionally. Typically, it's because the forester did something wrong, like put too much nitrogen on an already site that was fertile. But and occasionally you have outbreaks of it, and some sites are definitely more prone to it. But we don't breed for it, and one of the reasons why we don't breed for it is you can have things be a problem, but breeding a cycle takes a long time. So you
mhm.
can't just stop everything you're doing to try to add another trait in, unless it's really going to result in good output. The genetics of the pathogen is an active area of research. There's been a lot of great work done by U.S. Forest Service scientists, um, folks at University of Florida, folks at our program to characterize the diversity in the pathogen, to identify how come sometimes a tree, um, the infection overpowers its resistance. We may have a family that we know tends to have resistance, um, lower infection than the mean, but how comes still some trees get infected? Looking at the pathogen side to see if there's genetic variation or basically marker variation associated with virulence. This is some of the findings from those is that the pathogen is pretty diverse. There are sort of distinct races, but so if you compare fusiform rust from North Carolina to that from Alabama, they're not going to be as related to each other as if you compare fusiform rust from Alabama to Georgia. But there's a lot of gene flow among the populations, and that gene flow really helps us out, right? That prevents a particular um, location from becoming so different that uh, we have to do more intensive testing there to make sure that you got to make sure you got loblolly pine resistant to that Alabama rust. We don't have to worry about that because there's gene flow among the populations. We have a particular study right now to understand virulence in the pathogen, to see why it's overcoming what we think are resistant genes. But the truth is, we haven't given as much attention to the genetics of the pathogen because there's so much gene flow across the region. What we find is if you just progeny test and plant trees in that region and just observe data on what's getting infected, what's not getting infected, at the end of the day, that's how we're going to get resistance in our population. Don't get me wrong, it's fascinating research, but at the end of the day, I got to answer to my stakeholders, and they want resistant pine trees. So we can't get too distracted.
Yeah, it looks like if there's any more questions. Looks like Angus is answering one of the questions in the chat. Um,
I got a little overexcited and answered one about how big the pine genome is, Trevor. You might want to correct me if I'm wrong on that one. 7 times the human genome.
Yeah, several times. It's 22 gigabases. I don't know, Jared, do you know what the human genome is?
it's 7 gigabases, so it's like, or is it three? I don't know. I remember it's like something like seven
The
times the
pine
size of
genome
the.
is 7 times larger than the human genome.
Yeah,
It's
right. So
amazing.
it's
Yeah.
3 gigabases human.
And
Uh,
it's highly repetitive. And what does all that mean? Pine trees have been around for a long time, very, very long time. They've been evolving and having mutations. One of the mutation that happens is the DNA basically, when you're writing in Word, your manuscript, and you write the same word twice, it says delete repeated word. Or you made though that's a very common mutation. And when I say common, I mean it's happened many times over the hundreds of millions of years that pine trees have been around. And it makes it very difficult to assemble the genome because you get your data back. It's like these pages in a book that tells a story, but all the pages are mixed up and you have to put them in order. And the problem is like 99% of the words on every page look the same. So it's very difficult to put that story together.
Yeah, so you just doing whole genome sequencing on a pine is a big, very expensive ordeal. So having a marker panel is what saves you on for that genomic selection. Is you're only doing those whatever 50,000 markers in every
Yes.
tree, and you're representing the entire genome with a sample of those 22 billion bases. You're only sampling 50,000 100,000 of those, but that is enough to represent um, the genetic recombination landscape in that population, right? Is that kind
And
of,
the way it works
yeah.
is that you have a population of trees that you know a lot about, and then you genotype those. And so you associate those markers in that population. So, for example, a big family that all has the same mother and father, if you genotype with those markers, a big family that all have the same mother and father, and you observe how the markers are segregating in there, you can say, hey, all these markers are on the same chromosome because they segregate together. Or these markers are definitely on different chromosomes because they segregate completely independently.
Yeah, just I was Googling the largest genome in the world belongs to a fern, and it is 160 billion bases. So pine is 20 billion bases. Humans are like 3 billion bases. Chestnuts are 700 million bases. So it kind of gives
Okay.
you a sense of like the orders of magnitude and genome sizes. And
And also the evolutionary
yeah,
history, right? Ferns were some of the first plants,
yeah.
and
So
pines before chestnuts.
yeah, and that is a major limiting factor. You can't like with chestnuts, we can sequence a chestnut genome with maybe like $100. For pine, it's a
So
lot.
what?
Yeah,
So Jared, the next step is you got the genome part down. It's having that population of real trees, right, and experimental designs that now you start
yeah,
associating. And I know you've done excellent work, showing the quantitative nature of a resistance. That it's not chestnut trees don't just have this one gene that makes them resistant. They've got a whole evolutionary biology of genes of mutations that have evolved to make them resistant.
cool. Looks like we're through the questions, and I really appreciate your time, Trevor, and presenting with us today. And it's very inspiring to see all the progress you've made over just using these what seemingly are simple techniques, but they're actually complex in the logistical implementation. And it involves a lot of people. And this is the same with chestnut. We need all of you to help us plant these trees, maintain the progeny tests, measure the tests, and so that's really a group effort. Breeding is a major group effort. And so everyone has a role to play in that. Our chapters and the people that have volunteered with us it's very important to maintain that work going forward. That's essential and it works. So
Yeah,
this
if I could
shows,
just emphasize,
yeah,
Jared, if I could just emphasize, I showed our team of people. We got 4 full-time research associate staff that help us implement the breeding. We've got those full member partners. The way that it works is we assign breeding assignments every year to those folks, and if something happens, they get busy with something else, and the breeding doesn't get done. It hurts all of us as a group. So staying organized is it's so crucial to keeping the cycle spinning. Also, I can put my email address in the chat if anyone has any questions they want to send me to follow up on. I'd be happy to.
thank you, Trevor. And thanks all for coming. It's really great to have Chestnut Chat again. We'll see you next month.
Thank you.
And also, I just want to say, keep an eye out for the November 21st Chestnut Chat. Jared, do you want to make a short pitch about what's happening in November?
So Jessica Rutkowski, she is a breeder, small grains breeder, but I originally saw her paper it's like called The Practical Guide to Genetic Gain, and it really, to me, she was able to communicate very beautifully the basics of breeding and quantitative genetics and then the use of how these technologies like genomic selection and high throughput phenotyping layer on top of a solid foundation of doing really good field trials, good cross designs, keeping good records of your breeding program and the data analysis and everything. So she understands what is required for success very well, and I think she's a great communicator on that front. So I'm really looking forward to hearing from her. It'd be similar vein to what we heard today with Trevor, just in another context. So yes.
Am muted. Thank you so much, Jared, and thank you so much, Trevor. So please tune in for our next Chestnut Chat on November 21st. And you can find the recording for this on our website on the Chestnut Chat archive. It'll be posted early next week. Thank you all and see you next time. Have a great day.